Just like we had to talk a little bit about chemistry to understand the biomolecules, we also have to talk a little bit about thermodynamics to understand how energy restricts what living organisms can do. So chapter 8 is about energy on the first part and about enzymes for the second part. So we need energy to do all kinds of different functions in our cells. When we talk about a cell's metabolism, that's all the different kind of chemical reactions, both those that use energy and those that release energy. And just like anything else in the universe, organisms are subjected to the laws of thermodynamics. When we think about metabolic pathways in organisms, it's usually a series of chemical reactions where some starting molecule gets acted on by some enzyme that produces an intermediate that gets acted on by another enzyme that produces another intermediate, and so on and so forth. And for a lot of these pathways, they're not actually perfectly linear. They're often branched. And a lot of metabolic disorders are not caused by never getting to D, because there's different ways to get to this final product, but rather it can be a buildup of some of these intermediates that are toxic. So for example, if there was a mutation in enzyme number 2, you wouldn't make C directly. And if this was the only way to get to D, you wouldn't make D either. But the other thing that happens is that this B would build up because A would still get converted to B by enzyme 1. And it's a buildup of these intermediates that are sometimes toxic in metabolic disorders. So when we think about different kinds of chemical reactions, we can think about those that use energy and we can think about those that release energy. And we're going to see there are several different vocab words throughout this chapter that really relate to those two different concepts. So pathways that are catabolic are going to break things down. So that's going to be taking things like cellular respiration, where glucose in the presence of oxygen reacts to form water and carbon dioxide. So we have this large complex molecule that gets broken down into these simpler molecules. Catabolic reactions release energy. And so that's what the purpose of cellular respiration is, to release the energy that's stored in our food and pass it to ATP so our cells can use that energy to do work. On the other hand, anabolic pathways build things up. So when you think about anabolic steroids, they're used to build muscles. Okay? Anabolic pathways consume energy. So this could be building any structures, whether it's muscles or parts of the cell or protein or DNA or whatever. We take this same chemical reaction of cellular respiration and run it in reverse. So if we add energy to water and carbon dioxide to form sugars, that's photosynthesis, and that's an example of an anabolic pathway. What energy is the capacity to cause change. So doing any kind of work requires a source of energy, whether it's just maintaining the cell's structures, moving things around, replicating cells. It takes a form of energy. And there are two basic kinds of energy. Kinetic energy is energy associated with motion. So we typically think of, in physics, the example of the ball rolling down the hill as kinetic energy. In biology, the relevant form of kinetic energy is heat energy or thermal energy, the energy associated with the motion of atoms or molecules bouncing around. The other kind of energy is potential energy, energy due to structure or location. So the classic physics example is the ball sitting at the top of the hill. It has potential energy due to its location. What's most relevant for biology is energy stored in chemical bonds is a form of potential energy. And the energy stored in chemical bonds can be released as heat. The energy in that ball sitting at the top of the hill can be released when it rolls down. And the study of these energy transformations, converting potential to kinetic and kinetic to potential, is called thermodynamics. And the laws of thermodynamics govern everything in the universe, including organisms. The first law of thermodynamics is the conservation of energy. The amount of energy in the universe is constant. Energy cannot be created or destroyed. It can be transformed, it can be transferred, it can be changed from one form to another, but it's not broken down or used up even when we use it, right? So the power plant is not creating energy. It's converting energy in coal to energy in electricity. When you go for a run and you get home and you're really, really tired, all the energy that you used isn't gone, it's just dissipated as heat energy. In biological systems, the source of energy is the sun, and it gets fixed into organic molecules in the process of photosynthesis, and then when organisms use that energy up, it dissipates as heat energy. The second law of thermodynamics is the law of entropy. The law of entropy says that energy transformations increase the disorder in the universe, or the entropy. So when we convert the potential energy in our food to the energy that's in ATP during cellular respiration, some of that energy is lost in that conversion process. It's lost as heat. So the energy is not gone, it's dissipated into the rest of the universe. And that heat energy disorganizes the air molecules around it. It makes it more disordered or entropic. So energy transformations increase the entropy or disorder of the universe. One of our characteristics of life from chapter 1 is that organisms are organized. We keep our structures in a particular place in a particular orientation. But living organisms are still bound by the laws of thermodynamics. They keep their local structures organized by expending energy, and that energy that's expended is released as heat energy that disorders the rest of the universe as that heat energy is released. If we think back to those different kinds of metabolic reactions, catabolic and anabolic reactions, those that release energy we describe as spontaneous. Now, spontaneous doesn't mean fast. What it means is that the reaction proceeds without an input of energy. Energy is released by this process, okay? And for a process to be spontaneous, it has to increase the entropy or disorder of the universe, okay? We can measure the change in energy in a system, whether the energy is increasing or decreasing. And that change in free energy is the Gibbs free energy is represented by this delta G. The reaction is spontaneous. If it's releasing energy, then our change in energy in the system is negative. So if we have a negative value for delta G, we are losing energy, we're releasing energy, and that's a spontaneous reaction. On the other hand, if we have to add energy to the system to get it to go, then we have a positive value for delta G, and that's a non-spontaneous reaction. So the release of energy happens when the system is unstable. So if you think about that ball at the top of the hill, just a little poke will convert that potential energy due to location into kinetic energy of the ball rolling down the hill. When it gets to the bottom of the hill, it's released all of its energy and it's stable again. The point of maximum stability is called equilibrium. When you're at equilibrium, you're not changing free energy anymore. And if you're not changing the amount of energy in the system, no work is being done. And if that happens in a living organism, if everything in a cell goes to equilibrium, then you're dead. So we can think of a couple of examples that go along with this. So here we have a guy at the top of a diving board. He's the same as a ball at the top of the hill. He has high potential energy, but he's unstable. And if he jumps off, he converts that potential energy to kinetic energy. And at the bottom, he has low free energy. The same thing happens if we have a drop of dye that we drop into a beaker. Initially, the molecules are close together and they have higher potential energy, but as they diffuse and they spread out all throughout the beaker, now they're as far away from each other as they can get. They're still bouncing around, they don't stop moving, but since there's no net change in direction, no work is being done. The same thing happens with chemical reactions. If we have a glucose molecule that's in this complex, high energy state, and we reorganize it into these smaller, more simple molecules, the energy is released. A couple more vocab words that describe these same situations. So if we start with some starting material like glucose that has high potential energy, and we convert it into these smaller, more simple molecules, the energy is released. So we're going from high to low. We have a negative value for delta G. Energy is being released. And so a reaction that releases energy can be described as exergonic if it releases energy, and specifically exothermic if it releases energy in the form of heat. So if you have those hand warmers for going to football games in the fall, it does a chemical reaction that releases energy that warms your fingers up. On the other hand, if you start with low energy reactants and you have to add energy to make the high energy unstable products, we have a positive value for delta G. We have to add energy to get this system to go, and so this is not spontaneous. So when we have to add energy, we say that that's an endergonic reaction. And if we have to add heat energy to get it to go, we say that's an endothermic reaction. It's absorbing heat energy from the environment. And that's how those cold packs work. When you break them and it does a chemical reaction, it absorbs energy and it makes the surrounding environment feel cold. Now this is important for biology because the energy that's released by exergonic reactions can be absorbed by endergonic reactions. And so we can use the energy that's released from one set of reactions to drive other reactions that our cells want to do. That process is called energy coupling, and the molecule that accomplishes this is ATP. Think of energy coupling as this model where we have a reservoir of water that turns a turbine that lights a light bulb as the water pours into this lower reservoir. If we had a closed system, as soon as the water ran down, we'd reach the point of equilibrium and no more work could be done. But living systems aren't closed systems. Living systems have to have an input and an output of energy. And as long as the tap is on and the water has somewhere to run to, we can keep this turbine spinning. But we have to have a source of energy, our food, okay? And we have to be able to release that energy. And if we can link these reservoirs together, we can actually release the energy in little bits that our cells can harvest and use to do work. So the molecule that does this energy coupling is ATP, adenosine triphosphate. So ATP is an RNA nucleotide. It has a nitrogenous base, a 5-carbon sugar, and 3 phosphate groups. It's adenosine triphosphate, okay? And energy is released when the high energy form of that molecule is reorganized by hydrolysis into a lower energy molecule. So this is what ATP looks like. We have our nitrogenous base. We can see that it's ribose because the 2' carbon has a hydroxyl group on it, and it has the adenine base, and it has 3 phosphate groups. When we add water to this molecule, it breaks off the terminal phosphate group and forms the lower energy molecules of adenosine diphosphate with just 2 phosphates and the free inorganic phosphate. Now, here's a confusing concept. It's not the breaking that releases the energy. It's the formation of the lower energy bonds. Breaking bonds actually requires an input of energy. Just like if you chop a log of wood apart, you have to use energy to break it apart, right? So it's not the breaking, it's the formation of the new bonds. So what actually happens during hydrolysis of ATP is we break the bonds in ATP and we break the bonds in water, and we reorganize them into ADP that has a new covalent bond between the phosphate and the oxygen from water and the hydrogens from water and the inorganic phosphate. So it's not the breaking part that releases the energy, it's the formation of the lower energy products. Hydrolysis of ATP, it actually does require energy to break the bond in the terminal phosphate group. However, the amount of energy that we have to put into the system to break it apart is less than the energy that we get back out forming the products. So it's a little bit confusing, but formation of the lower energy products is actually what's catabolic, actually what releases energy. Breaking things apart requires an input of energy. So when we think about cellular respiration, we do have to break our sugar apart, but it's not breaking it that releases the energy, it's the formation of the lower energy products. Simply put ATP in a glass of water, it would go through hydrolysis and release that energy as heat and light. But in our cells, that energy is transferred to target molecules. That phosphate group doesn't immediately form inorganic phosphate. It forms a phosphorylated intermediate of some protein that folds it into a different shape and allows it to do work. Once ATP is hydrolyzed into ADP and PI, it can be recharged by energy from other exergonic reactions. And so it can be recycled and the process continues.